I'd like to introduce our first speaker, Dr. Sandra Ramirez Arcos, who is a development scientist here at the Center for Innovation. She's an expert in bloodborne bacteria and how they form biofilms in blood components. And Sandra, would you like to come over?
Good morning and good afternoon, everyone, depending where you are. Thank you, Everett, for the introduction. What I would like to do in the next 10 minutes is to walk you through the process that it took to implement a new algorithm for bacterial testing of platelets based on the extension of the platelet shelf life from 5 to 7 days. So what we would like to do today basically is to discuss this process and not only to know the process, but also to know what are the gains on the safety of platelets based on the implementation of the new testing algorithm. Some of you may know that this is the system that we use to screen platelets for bacterial contamination, called the Bacteria Alert system, and it's basically a shaken incubator. This is a very high-level diagram of the process, and based on the interest of time, I'm not going into too many details, but it's important to know a few concepts here for the next slide. So basically, we take a sample of the platelet product, and it's inoculated into culture bottles that are shown here on the second graph. These bottles have a sensor at the bottom, and if there is a change in the pH, a decrease in pH, this sensor changes color from bluish to yellow, and this is what is detected by the Bacti system. If there is a change in color, then we'll have a positive bacteria alert result. It does not mean necessarily that we have bacteria there. It has to be a follow-up of those culture bottles that can be done through subculture onto agar plate or microscopy. And at the end, there are two possible results that we have of interest for this presentation. One is that we confirm there are no bacteria in the platelet product despite a positive bacterial result. And this is what we call a false positive. The second scenario that is important for us is when we confirm that the platelet product that was tested was contaminated with bacteria, and we call this a true positive. So what are the changes that we implemented since August 2017? The first one is we delay sampling by 12 hours. Now we are taking a sample of platelet products for inoculation into bacterial cultures after 36 hours of blood collection. We are also doubling the volume that is inoculated into culture bottles because we are using both aerobic and anaerobic culture. In the case of double apheresis, we are actually now inoculating 4 culture bottles. And also something else that is quite important is before the extension of platelet shelf life to 7 days, we were not quarantining the platelet units after sampling. Now we are quarantining them for 6 hours before they are being released into inventory. So how these changes actually can increase the safety of the platelet product? Here in this diagram, what we can see at the left is a figure of a platelet unit, and these green dots represent bacteria in very low concentration. Because we are increasing the volume, now we have the possibility to increase the chance of capturing those bacteria. Not only that, since we are increasing the time before there are samples, we actually giving bacteria the chance to replicate, to increase the numbers. And again, we have more possibility now to capture contaminated units. Now to the data. Can we confirm that these predictions are true or not? In this slide, I am showing results of testing of Buffy-Coat pool before extension of platelet shed life and after from August to November. I want you to focus on this row of true positive results. That means these are units that we confirm that they were contaminated with bacteria. You can see that the rate per 10,000 units has increased 5 times. So we know for sure that we have increased the safety of platelet products. Basically, this means that we are capturing more contaminated units. The organisms that are contaminating these units are mostly from the skin flora, but we have also captured some pathogens like Staphylococcus aureus or Streptococcus. Similarly, for apheresis units, we have doubled the detection of contaminated units. The difference in the rate between apheresis units and pools are due to the number of platelet concentrates tested. But then the take-home message here is that we actually have increased the safety of our platelet supply. So what happens? This is not a magic bullet, it's not foolproof, and unfortunately, despite all of these improvements, there is still a residual risk. How is that captured? Twofold. The first one is we still do quality control sterility testing of outdated platelet components. It's done in platelets that have expired in the shelf life, so they are between 8 to 10 days old. And since August 2017, we have captured 1 contaminated pool. Similarly, we unfortunately had 1 confirmed transfusion reaction. As you can see here with these very small numbers, only for 3 months, the rates seem to be very high. I think we have to wait few years before we can see that in reality, there is a residual risk, but it's less than before we extended platelet shed life. So, did these results take us by surprise? Not really. We ran a development study last year, and what we did using 4 different organisms as a model organism for this implementation, we spiked intentionally contaminated platelet units with 2 gram-positive organisms that are considered to be common platelet contaminants, and they grow very slow, and 2 gram-negative organisms at the bottom of this panel that they grow really fast. In blue, you have showing what happens with the previous algorithm, that we were sampling platelets 24 hours post blood collection and only inoculated 1 culture bottle. In red, you have the new change. Now we are sampling 36 hours post blood collection, and we are inoculating 2 culture bottles. As you can see with the gram-negative organisms, Serratia, marcescens, and Klebsiella at the bottom, now we have increased the safety, what is predicted at almost 100%. However, with the slow growers, even that we have now increased the chances of capturing contaminated units, there is still a gap, and this is inherent to the process. There are organisms that grow very, very, very slow, and they are in low numbers in the platelet concentrate, and they will still escape detection. So what is next? What is the next big change? I'm happy to report that very soon, hopefully in the next 3-4 months, we will be able to implement this change. So what is happening until now is that any associated component to a platelet concentrate that has a positive bacterial layer result is discarded. And as I mentioned in one of the first slides, a positive bacterial layer result does not mean that the unit has bacteria or not. So now we will be implementing a follow-up of those culture bottles in-house that will allow us to release co-components of units for which the results are false positive. So if we have a positive bacterial bottle and we confirm that there are no bacteria in that bottle and that the unit is not contaminated, we will be releasing at least 4 red blood cells and 3 plasma units associated to a platelet pool, as an example. So to wrap up, I can say that the implementation of the new bacterial testing algorithm has increased platelet safety. It's not a magic bullet, and we will still have residual risk. And we know that with the upcoming implementation of the in-house follow-up positive bacterial culture, we will be able to release co-components and, of course, decrease wastage of products that otherwise can be used for transfusion of patients in need. And with this, I would like to acknowledge a lot of people who have been involved in the implementation of 7-day platelets.